Hello, and welcome to this week's episode of I Came for Science, a show about sex and reproduction and inclusion in STEM. I'm your host, Laura Surratt. I'm a professor of biology, and I've been studying animal sex for the past 30 years. Please join me as we normalize talking about sex and what we know about it, as well as the journeys of the scientists who study it. In this week's episode, we're going to talk all about penises. We'll explore the wide variety of penises found in animals and hypotheses for why they evolved. My guests today are Dave Hosken and Matt Dean, two biologists who study the diversity, evolution, and function of animal penises. Dave Hosken is a professor of biology at the University of Exeter in the United Kingdom. Welcome, Dave.
Great to be here, Laura. Thank you very much for inviting me.
I'm so glad you're here. I've followed your work for a long time, and I'm happy to have the chance to share it with others. I'd like to start by talking about the diversity of penises found in the animal kingdom. So what are some of your favorites?
So before I get to some of my favorites, you've just hit on it exactly why the penis is a kind of interesting character, is that its job is really simple. It's to take sperm from a male and transfer it over to a female. That could just be a simple tube, right? It doesn't, but the
doesn't need to be fancy.
Yeah, it doesn't need to be fancy. But exactly as you say, penises across the animal kingdom are absolutely crazy in their diversity. And as an evolutionary biologist, that for me is really interesting. Why is something so varied when it could be really, really simple? Why are species that look so much like each other generally, you couldn't tell them apart just to look at them? But if you look at their penises, you can say, oh, that belongs to species A and that belongs to species B. There's something really weird about penises.
Yeah. So what you're saying is not only are they more complicated than they need to be, but they're more different from each other than you would expect.
Exactly right. Yeah. Exactly right. And so that's why I came at the penis, if you like. There's nothing more nefarious in it than that. It's a really bizarre trait. What's going on? So to your question, and by the way, thank you very much for the very kind words about following my work. Appreciate that very much. As a scientist, you don't often get to hear that face to face, so I very much appreciate that. I think there's two penises that are super interesting. One of them actually is exactly the opposite of the complexity. We were just saying penises are really complex. This one's super simple. It's the hypodermic syringe penis of bed bugs.
So weird.
Yeah, it's very, very cool. And I'll return to that in a minute. And another one is it's a female penis. So if a penis is a device for transferring gametes from one sex to the other, there's one example I know of. It's a cave-dwelling insect where the female has the bit that goes inside the male to collect the sperm from the male and bring over to her. So that's kind of interesting too. The hypodermic syringe that's involved, that bed bugs and actually other insects have, that I find really interesting. It really is like a hypodermic needle, and males stab females with it, oftentimes not using the female genital tract, so not putting sperm into the vagina, shooting the sperm in through the side of the body wall. And the reason we think that's the case is the closer you can get your sperm to the eggs, the greater the chance you have of winning the race to fertilization. And the males that have this hypodermic penis basically circumvent, or are trying to circumvent, female control over who fertilizes their eggs, but also just getting the sperm right in there as close as they can to the egg. So they literally puncture the female body wall in some instances and ejaculate the sperm straight into the female's body cavity. That's pretty weird.
So it is weird. And I guess I haven't really thought about this before. Like, I knew about that case, but if that's the situation, I always think sperm need to enter a particular way to be able to get to the location where they fertilize females' eggs. Is that different in the bed bugs?
I think what they've done is they've just moved the site of fertilization.
Okay.
But what females have done in response to that in some groups of bed bugs is they've basically evolved a secondary reproductive tract.
Sort of entry to the reproductive tract, is that what
Yeah. So they've got a little groove on their underside that guides males into another opening in the female body cavity, so that females don't get hurt by the stabbing. So males have sort of evolved this thing to try and seize control over fertilization, and females have struck back and said, no. You'll do what we need you to do, and you'll use this organ again. So it's kind of Interesting.
Yeah, very interesting. I think some of my favorite crazy penises are, I mean, of course, there's the duck examples where the males have these corkscrew penises
Yep.
and the
Yep.
females have a reverse corkscrew. And so that's wild. What got me interested in penises was damselflies, though, because for my master's, I studied damselflies. And there's this classic study by Jonathan Waggy about penises that aren't just used for transferring sperm, right? They're used to also scrape out sperm from previous males. So female insects can store sperm and sperm storage organs, and then males have evolved penises that are able to go in and scrape out the sperm of previous males and then insert their own sperm. And it's wild. The diversity of penises and damselflies just blows your minds.
Yeah, that's one of the first studies that I saw too. And
Yeah.
it was during my grad program, and I just got really interested in sperm competition. And there's a whole nother story there about testis size evolution. But seeing that, it's like, whoa, this is really mind expanding stuff. And
Yeah.
I seem to remember there was a wonderful electron micrograph showing
Yes.
the backward facing spines on the penis with rival male sperm. And
Yeah.
if you think about it, it's a pretty neat trick. A female's mated with somebody else. You want to fertilize her eggs, so you just scrape out, all the rival male's sperm. It's fantastic
Yeah.
and a really neat solution to a kind of bit of a tricky problem.
Yeah.
It's
Yeah.
good, very good.
Yeah. And it's not to say that the females don't have any agency. There's lots going on in the female reproductive tract also that prevent males from transferring sperm or allow them to
Yep,
eject sperm, all sorts of cool things. We'll get
yep.
to that later. So now we've established that there's lots of diversity in penis shape and size and structure. But what I wanted to talk to you about today was really how this diversity comes about. So I was wondering if you could kind of take us through your thoughts on that.
Yeah, I think it's generally accepted by everybody that the general answer to that question is sexual selection, more strictly post-copulatory sexual selection. And I think the most general explanation is cryptic female choice. So just to unpack that a little bit, post-copulatory sexual selection, really what I mean by that is the sexual selection that occurs after intromission. Male inserts his penis and there's a whole episode of selection there. Females have made their mate choice. They've gone, okay, you're the male I want to copulate with. Males copulate, and then the things that happen during the process of copulation and subsequently where females and male competition decides who actually gets to sire the offspring. So cryptic female choice is the name we give to mate choice that occurs after intromission, and that seems to be the most general answer to penis evolution. It's basically, what can I do to stimulate the female in the right way to make her use my sperm? Now, the case we were just talking about was a case of male-male competition, sperm competition with the damselflies and the removal of sperm. That clearly plays a role in the evolution of the penis, but I think most people would accept that it's female choice that's the main driver of penis evolution. And there's some great work done by Bill Eberhard, who's an absolute hero of mine,
Me
on
too.
this. Yeah, he's very cool
Yeah.
and a fantastic man as well, on the sorts of things that males do during copulation with females. They're waving their legs in front of them, they're tapping them in interesting ways, moving parts of their bodies in certain stereotyped ways. And all of that sort of copulatory behavior doesn't make any sense until you start to think about it as a way to stimulate females so that your sperm will be used by the female to fertilize her eggs. And, just to be absolutely clear to all the listeners, males can only have evolutionary fitness. The single most important thing in the evolutionary traits, they can only have reproductive success through females. So females' eggs really are the what's the word? The grand prize. So anything you can do as a male to access those eggs, will be selectively favored.
Cool. Thanks for explaining that. Now, I know when we first talked about this interview, you were explaining to me that you saw this all in the broader context of the evolution of sex and how that ultimately leads to that process. I was wondering if we could back up to talk about those issues as well.
Yeah, no, that's great. So things don't have to reproduce sexually. Lots of things, can clone themselves. But most of the stuff that we see when we look out the window, most of the animals and most of the plants produce gametes produce sex cells, and those sex cells combine to form a new individual. Why do they do that? What does sex do? Well, it mixes up genes. So if you reproduce without sex, you basically produce copies of yourself with some variation, but a small amount of variation. Once you start to have sex, then you can produce offspring that are very different from the parents. Now, there's lots of reasons why sexual reproduction might be favored. It's still, I think, one of the great unanswered questions is
Yeah.
exactly why most things are sexual.
Mm-hmm.
We've got answers, but they don't always outweigh the costs of sex. So
Right.
we
And they
can
don't
sort of
explain all the cases that we see
yeah,
out there.
yeah,
Yeah.
yeah. So
Yeah.
I think we just park that to one side for now because that's a big one. But
Yes.
once you've got sex, once you're reproducing sexually,
Mm-hmm.
it seems that almost inevitably in complicated organisms, so not in bacteria, but in bigger things like humans and trees and insects, once you're reproducing sexually, you inevitably end up with sexes, the biological sexes.
Mm-hmm.
And what I mean by that is you end up with things that make big sex cells, eggs, and you end up with things that make small sex cells, sperm. Sperm producers we call male, egg producers we call female. And solving that problem, why there's this magic number of two, is also a fantastic biological story. Jeff Parker was most influential, I think, in solving that particular problem. And basically what happens is the sperm producers evolve to parasitize the egg producers' eggs.
Mm-hmm.
So males are the ultimate parasite in a sexual system in some sense,
Mm-hmm.
which is kind of interesting to think about that as well. And two's the magic number because if somebody starts making smaller gametes than sperm,
Mm-hmm.
than the current
The,
sperm
the
producer, the organism that makes the big sperm gets outcompeted by the one that makes even smaller sperm.
because those are more parasitic. They're less costly to make. Is that right?
You can make
Or
more of them.
you can
It's
make more
you've got
of
to
them.
yeah,
Yeah.
you're cutting a pie up into smaller
Mm-hmm.
and bits. You've got
So you have
more
more
bits of
sperm.
pie,
Mm-hmm.
right? So two's the magic number because of that fundamental
Mm-hmm.
competition. And eggs can't get smaller because if you combine small and small, you don't have enough resource then the developing baby, the
Mm-hmm.
offspring, the zygote to survive.
Right.
So
Mm-hmm.
eggs kind of get trapped at some size once the small gamete producer, the sperm producer, evolves really small gametes. Did that make sense?
Mm-hmm.
So then we've started off with, okay, we need to have sex.
Mm-hmm.
We end up with the sexes through what can be thought of as the primordial conflict between maleness and femaleness.
Mm-hmm.
Once you've got sexes, then all sorts of secondary conflicts between males and females evolve. And that is an absolute Pandora's box of things to talk about, but more than happy to as well. That's great, I think.
Yeah. Well, it's been a pleasure. Thank you so much for joining us today. And I look forward to talking to you again soon.
Yeah, brilliant, Laura. Thank you so much for the invite.
Thanks so much, Dave. Next, I will continue our conversation about sexual conflict and the biology of animal penises by talking with Matt Dean, who's an associate professor of biological sciences in the Department of Molecular and Computational Biology at the University of Southern California. Welcome, Matt.
Thank you for having me.
Yeah, we're so happy to have you. Can you start by describing what general area of reproductive biology you focus on?
Sure, I'd say the biggest umbrella is sort of this concept of conflict. So there's in reproduction a lot of conflict between males and females. And in my early postdoc years, I got really interested in proteins of seminal fluid, kind of how they were playing a role in mediating this conflict, some questions that, quite frankly, you were interested in too. And that led me to study this structure called the copulatory plug, which is basically coagulated seminal fluid and probably functions in the context of sexual conflict, which is, in that case, a male's way of securing his reproductive investment. And one thing led to another, and I don't really know how I got here, but now I study bacula, which are bones in the penises of many mammal species, and they're sort of remarkably diverse and have captured my attention.
Yeah, it seems like a lifetime of research ahead of you on this
Yes.
topic. Yes. So previously on the podcast, we talked to David Hosken about invertebrate penises and how wonderfully wild and diverse they are. So I was wondering if you could tell us a little about the diversity of penises found in vertebrate animals and what some of your favorites are.
Yeah, I mean, I would think I know Dave pretty well, and I hope he showed you a picture of the seed beetle. But one of the interesting things is that it sort of doesn't matter what group you're studying. So insects or fish or birds, mammals, if there's internal copulation, there's almost always very, very rapid evolution of genitalia. And so all the things that Dave talked about probably also applies to mammals. Their genitalia are incredibly diverse. I sort of use this, it's hard to do over just audio, but if I'm giving a talk, I sort of have these pictures of all these primates on a slide,
Mhm.
and many of the audience members are not mammalogists or maybe not even scientists, and yet they can recognize them as primates because we know we have a search image for it. But if I showed you images of their penises, no one knows what anybody's looking at. Everything's as different from everything else as, it's just a very confusing amount of morphological divergence. What are my favorite ones? I don't know. So I sort of like cetacean penises. We have a paper on the evolution of cetacean pelvic bones. Cetaceans are whales and dolphins.
Mhm.
And they're very dexterous with their penises, so they can kind of operate,
Yes.
they can move them around. And we studied the bones of where those muscles attach that operate the penis. And the analogy is like if you've ever seen a person in the park flying a trick kite, they have two lines
Uh-huh.
going up to the kite, and they pull one and the kite will turn to the right, and they pull the other, the kite will turn to the left. That's how a whale or a dolphin controls its
Wow.
penis. We're basically studying the connections of those strings, but in cetaceans. So that's a cool one. I mean, they're all cool. So galagos have a particularly interesting penis. They have spines all over them.
Can you explain what those are,
What the
galagos?
spines? Oh, what
No,
galagos are? Let's see, it's a primate. What's the common name? Bush baby, I
I think
think.
that's
It's
right.
a
Yeah.
little primate with gigantic,
Huge
cute
eyes.
eyes.
Hmm.
Probably in some Disney movie somewhere, but it's a basal primate, and they have spines all over their penises. And that one's particularly cool because not my lab, but another lab figured out some of the genetic basis behind that, what puts spines on penises.
So that's like the bean beetle, the
It
invertebrate
is, yeah.
version. Yeah,
Yeah.
that's very cool.
Yeah.
Very neat. And I know specifically you've been focusing recently on the penis bone, the baculum. So I was wondering if you could explain to our listeners, like I think most people probably don't think about the penis having a bone. So explain what a baculum is and what species have it and what species don't have it.
So it's a bone in the penis. About 85% of mammal species have one. Humans are unusual because we don't have one. But for example, chimpanzees, our closest living relative, does have one, and most primates have one. There are a smattering of 5 or 6 species of primates that have also lost their baculum, but the ancestor of primates probably had one. If you look across the mammalian tree, as far as we know, every rodent on the planet has a baculum. Most carnivorans have a baculum. So carnivorans are things like lions and tigers and bears and weasels. And so there's a few exceptions there. Hyenas lost their baculum. Hyenas and a couple of their related species lost their baculum, but we're pretty certain the ancestor of carnivorans had a baculum. Bats are a little weird. There's a bunch of bats that have a baculum, a bunch of bats that don't. I'm kind of naming the big groups of
Yeah,
mammals.
no, this is great.
The biggest group that doesn't have one would be like the artiodactyls, or what some people call the set artiodactyla. So that includes things like antelope and deer and elk, and also whales and dolphins now turn out to be more, in that clade. None of those things have a baculum. None of the horses, so none of the perissodactyls, no horse-like things have a baculum.
What
And
about like animals, household animals like dogs, cats, and agricultural animals?
yeah, so dogs have a baculum. It's a pretty cool one. It's a sort of like a V-shaped, long V-shaped thing. Cats also have a baculum, but it's really, really tiny. And in fact, there's an interesting story in the literature. Saber-toothed cats, they've never found a baculum in any of the fossil remains. And
Really?
so, somebody in this field, Adam Hartstein, Stone Rose wrote, the title of the paper was like, at what point can we say saber-toothed cats don't have a baculum? And he makes the argument that we've got enough data now from different fossil remains. But anyhow, the point is that cats have a really tiny one. All your other, like, cows, sheeps, goats, none of those things have a
None
baculum.
of them have them. Okay.
Yeah, so I think that covers, yeah.
Very interesting. I was especially interested in your research about how the baculum evolved. And like when you're talking about this distribution of which species have it and then these unusual species that don't have it, I was wondering if you can start by telling us, like, before you got into this field, before you started doing your research, what was known about baculum evolution? And then what did
Yeah.
your research add to that?
So a lot was known about bacula because for hundreds of years, mammalogists have used, taxonomists, people describing species, have used the baculum to describe species. In many cases, chipmunks, for example, are a very famous example of this. There are a lot of different chipmunk species that look really, really close morphologically, and so they're very difficult to distinguish species boundaries from. But it turns out their bacula are wildly different. It's following the same pattern I told you before, which is sort of on any scale of evolutionary time, genitalia just seem to evolve much more rapidly than the rest of the body plan. And so a lot was known kind of about structure and, morphology. There would be like a lot of morphological drawings in the old scientific literature. There were sort of two things that were not really well known. One was kind of just compiling all the data into one study. And so that required, now it would be easy. We would just employ ChatGPT or something like that and have it read all the literature for us. But
That'd be
back then we
lovely.
had to read thousands and thousands of papers to
Right.
basically score which species had a baculum and which didn't.
Hmm.
We actually wrote some software to like help us read PDF papers and
Oh, wow.
bring us to the area of the paper that
You
we
were
thought
early AI.
we were early AI. That's right. And so, if I was at a conference or something and I would ask a legitimate mammologist, which I am not a legitimate mammologist, I would say, this baculum, this is a really weird structure.
Yeah.
It seems to be distributed sort of off and on across the mammalian tree. Do you think it evolved multiple times, or do you think it has like a single origin and then it was like lost many times? And sort of what explains the distribution? And in all cases, people would sort of like look up at the ceiling and scratch their chin and say, yeah, it must have evolved multiple times, I guess, if you think about how it's
Mhm.
partitioned on the tree. And so I'd say, oh, great, where's the study? Like point me to the
Right.
study. Well, nobody's done that study.
Ah.
So that was my gateway drug into just understanding the evolution of the baculum. It was a literature search coupled with some
Sure.
evolutionary analyses. It was quite fun. And
Hmm.
our main conclusion of that paper is no matter how you look at it, the baculum evolved, like it arose multiple times during mammalian evolution, which is totally wild and crazy. I mean, it still surprises me.
Yeah.
Because it's like
Why does that surprise you? Yeah.
well, it's like a complex structure, I guess. And so what our study has shown is that it like, it sort of blinks on and blinks off during evolution. And that led me to kind of wonder about the genetic pathways involved. Is that like a light switch just turning on and off? And the distribution of the baculum is really weird. In most cases, we don't really know why the baculum evolved. I mean, that's still a mystery is what its function is. But for some reason, 70 million years ago, there were like four big derivations of a baculum in independent
That's
lineages.
wild. Yeah.
Yeah.
Why at that point did we all of a sudden need a penis bone? Yes.
Yeah, while some groups just, didn't.
Yeah.
It didn't happen in some groups.
Right.
So, and the other kind of thing is like if you take all the species that have a baculum and you compare them against all the species without a baculum,
Right.
you sort of ignore evolutionary history and you just like make two groups of those species.
Sure.
There's nothing that separates them, at least not consistently. There's
Right.
not like everybody who has a baculum mates in this way and everyone without a baculum mates in that way. There are some patterns in some of the groups where you see like a correlation between something like baculum shape or size and features of mating system, but kind of the rule is that whatever you see in one group kind of doesn't apply to other groups. There's no
Mm.
like general
Right.
explanation for
pattern.
why it arose or how it.
Yeah. Even though they don't fit into those groups, what would have been your hypothesis for like what would have differentiated the groups with bacula and without?
That's a good question. Probably any hypothesis is overly simplified because
Right.
the baculum is not, it resides inside a penis. And so all the soft tissue anatomy is also going to play a role in whatever aspects of mating system. The mating behavior, the copulatory behavior is all going to play a role. I guess I would have thought that species where there's like a lot of multiple mating, where females are mating with at least more than one male in a single estrus cycle, and then as that kind of gradient goes up to like really, really high levels of promiscuity, I would have predicted something scales with that.
Mm-hmm.
Call it what you want, size, shape,
Right.
complexity, I don't know.
Mm-hmm.
But it turns out that, in some groups it does, but it's not a general finding. So in sort of evolutionary genetics, reproductive genetics, there is a inherent conflict of interest over fertilization rates and fertilization outcomes. And selection tends to favor males that speed up that process somehow, some way. And this could be selection at the level of behavior or the level of their seminal fluid or their sperm, phenotype, swimming speed, things like that. And on the other side of the equation, selection is going to favor females that control the process. This is often called female choice, which is probably not a great term to use, but if you allow me a little bit of anthropomorphism,
Sure.
females,
Yeah.
they have to make good mating decisions, especially in a
Right.
mammal where so much maternal investment goes into raising an offspring. She has to choose wisely in terms of her
Mm-hmm.
mates. And so the bottom line is that I would have thought the baculum plays some role in that process, either helping promoting sperm transport, female stimulation to get peristalsis going to help that
Mm-hmm.
transport, or to initiate that sort of next stage of reproductive investment in a female. And so that type of conflict is going to be much elevated in a promiscuous species. So I thought whatever the baculum is doing in
Right.
that context, it should be more dramatic in the
Doing it more in those. Yeah.
species. But
That's really
turns
interesting.
out not to be that simple.
Huh. So why did humans lose their baculum?
So I'm sure there's some disagreement in the field. I would say that no, there's nothing that is in common with species that have lost the baculum. There are plenty of statements in the literature that suggest, oh, humans lost their baculum because they walk upright. And it's like, oh, okay. But there's plenty of species that lost their baculum that don't walk upright. So I don't really understand that as an explanation. But basically, I would argue that no, there's nothing. Oh, hyenas are another one. So hyenas
Right.
have really crazy genital anatomy. The females are what's called masculinized. Their clitoris is like the size of a penis. And so they're the only carnivore. Well, actually, there's two groups that have lost their baculum. And so people say, aha, it's because they have the crazy genital anatomy of the females. And I say, okay, but there's plenty of species that don't have crazy genital anatomy that have also lost the baculum. So it's just,
There's
again,
no
it's
one explanation. Yeah.
there's no one explanation.
Gotcha.
And it just might be that, on top of the kind of rapid evolution in terms of morphological divergence of the baculum, its loss or its gain is also part of that process.
So
So
interesting.
yeah, I really don't know.
And so your research found that there were independent evolutionary events that led to bacula, and you had said you were interested in the developmental biology, the genetic mechanisms underlying that development. So what did you find there?
So in recent years, we've kind of taken the system full bore into the laboratory mouse. And the reason for that is there's tons of genomic resources. There's a lot of genetic engineering that you can do with a mouse. And so we decided to start to tease apart the developmental pathways that give rise to a baculum. Your question earlier about sort of what was known, the other thing that was known about the baculum is that, at least in a rodent, it develops after birth, and it is
Interesting.
highly reliant on testosterone.
Okay.
So there were a couple of experiments done 50, 60 years ago that either focused on genetically abnormal mice that the males did not respond to testosterone or androgen signaling, or, the sort of brute method is to castrate a male mouse.
Hmm.
In both of those cases, they will not develop a baculum. And so you might think, oh, that's a great tool to like understand what the baculum does, because I can just compare castrated versus uncastrated males and then then that's my baculum, no baculum comparison.
Mm-hmm.
The problem is the castrated males don't fully develop into a male. They're highly feminized because they haven't gone through they haven't gone through puberty correctly. They're not responding to androgen signaling.
Yeah, like in terms of experimental design, they weren't a good control because there were a lot of other factors that were different between them. Yeah,
Correct. Yeah.
gotcha.
And sort of, I'd say 5 years ago, or maybe even longer, my big pie in the sky idea was to genetically engineer a mouse that didn't have a baculum. And
Mm-hmm.
I thought it would take me 10 years, but I had this amazing postdoc. I had this amazing grad student who's now a postdoc, not with me, who made that 10-year plan a 5-year plan. So
Wow. What's that person's name?
Caleb Guillon, he's currently doing a postdoc at Caltech. So he's shout out to Caleb. He made my dreams come true. So what we did, we basically did like three things. So, and I'm giving you the three success stories on this ocean of failures. And so one was we had to figure out what androgen signaling, how it was actually working during the development of the penis. So we employed this technique called ChIP-seq, chromatin immunoprecipitation. And without getting into the weeds, it tells you where androgen is binding in the genome to turn on genes and do what genes do. And when we did that in a developing mouse penis, we saw all of this androgen binding near bone development, near genes that were known to be involved in bone development in other systems like our limbs and our fingers. And so that's kind of interesting because if you think back on the presence absence, the ultimate light switch might be androgen signaling.
Interesting.
Yeah, so like you either do it or you don't, and then that is an untested hypothesis, purely speculative, but one we're pursuing in other species. So that was one bit of data. The other bit of data, we employed a technique called single cell sequencing. And again, without getting into the weeds, what we can do is harvest a developing mouse penis and separate it into all of its individual cells and understand what genes are being expressed in all of those different cells. So as differentiation is occurring, we can see what's going on at the level of gene expression. And the bottom line there was we identified these cells that are called chondrocytes, which are cells that lay down cartilage in a structure like the baculum or your limbs. And so we could compare them to limb bones and we could see there's some similarities, yeah, but there's some differences too. And a big one seemed to be the expression of androgen receptor. So putting those two things together, it was sort of the first molecular insights into this pattern we already knew from a while ago that androgen is required to produce the baculum, but now we sort of knew the players. We knew the genes. Cool. And then the third big thing, which was a genetic engineering experiment, which is one of the main reasons we use mice, is we sort of put those two things together in a way where we could knock out androgen receptor. That might sound terrible because then the mouse would not develop into a male. Right. However, the trick that we used only knocks out androgen receptor in the chondrocytes. So it leaves androgen receptor alone in the rest of the cells that are going to determine male features, testicles, spermatogenesis, all the things associated with puberty. So we left all of that intact, but we knocked out androgen receptor only in the chondrocytes, and that will affect the whole skeleton. However, we were okay paying that price because there's not a lot of sexual dimorphism. There's not a lot of differences between males and females in terms of the rest of their skeleton. We knew, or we predicted based on the sensitivity and requirement of the baculum, that we would absolutely obliterate that. And it turns out that's what we did.
That's what happened.
Yeah.
Wow.
Yeah. And so we had now a mouse that lacked a normal baculum. There's a little bit left. It's not totally gone, but it's probably a quarter of the size of the entire thing, or maybe even less.
And let's just pause and think about why that's so important, because like you had this evolutionary analysis, and from that you couldn't really differentiate any selective forces that maybe were selecting for or against a baculum.
Right.
And even if you did, it would just be correlational, right?
Exactly.
And so am I correct to say that this would be an experiment where you could actually test what the function of bacula were for the first time? Yeah.
Yes, exactly. So you get a direct test of the baculum. It can always be true that whatever defects we observe are not actually due to the baculum itself, but something correlated with that we don't know about. So that's always true.
Right.
In order to fully understand or address a question like, what does the baculum do? Or what does X do? All geneticists take the approach of break the system and then build it back. And then you fully understand what it does. And so we're in the process, we've broken it, and now we're in the process of building it back. But we need money to do that.
Okay, so if you can share it, what have you found so far about mice without bacula?
Yeah, in fact, it was just published about a week ago in PLOS.
Congratulations. That's
Thank you.
awesome.
What we learned was the males that have an abnormal baculum, they can't copulate. But by and large, there are some
Uh,
exceptions which are interesting. We'll talk about that in a second. But by and
so they
large,
can't like get their penis into the females.
we don't exactly know where the failure is. What
Okay.
we know is they do not copulate to the point of ejaculation. And the reason that we know that is because in mice, when a male ejaculates, he leaves a copulatory plug. It's my
Like you were
previous
talking
slide.
about before. Yes.
Yeah.
Yeah.
And so it's coagulated seminal fluid that stays visible for 1 to 2 days. And so we can check females that are with these males. We can check them twice a day, once a day, whatever. There are no copulatory plugs. And that's in spite of the fact that the males are really interested in mating. They have totally normal mating behavior. They mount females. You often see like all the fur is matted in the around the female's genital area. So he's trying to copulate. Is our inference, but he can't, and so we don't know the reason why. The exceptions to this are some of the males. We sort of got lucky here. When I say we knocked out the baculum or we disrupted its development, it's not a black or white statement. And so some of these males were really, really, really messed up. They just had a little tiny sliver of a bone left over, and some of them were sort of less messed up. They were still very, very far from normal, but they were less messed up, and so they had like a longer bit of baculum. The site of the baculum that's kind of grabbed our interest lately is this bulb that's on the proximal end of it, and it serves as an attachment site for a muscle in a normal mouse. And so nobody knows what the muscle does. Nobody knows how the male moves its baculum during copulation. These are all experiments we want to do. But the fact is, there is a big muscle that's attached to the back of the baculum in a normal mouse. In the mice that are really messed up from our knockout, they lack that entire region of the baculum. And so we don't think there's any muscle attachment there.
Wow.
The ones that are sort of less messed up, they have the little sliver of a bone and some of that proximal bulb. And so what we think is happening is we've essentially kind of accidentally rescued the knockout a little bit. They had a little bit of muscle attachment. And while we don't know what they do, whatever it is they do with it, we think these males that are not super, super messed up, that they can still do that thing, whatever that thing is. And those are the only, I think out of, God, I could look it up, hundreds of crossing days. I think we only got 3 litters, and they were from the messed up males. And there were 2 males that had a relatively less messed up baculum.
Interesting.
If that makes
Like
sense.
an intermediate one. Yeah.
Yeah.
Very interesting. Great. Well, thank you so much for your time. I really appreciate it. And thank you all for listening. Thanks also to our production team, Isabel Espinoza, Lane Patton, and Daphne Trilliana. The music used in this episode is from I Don't Smoke by the Mythical Score Society.